Now, let's continue with the vesicular transport. Guys, first of all, vesicular transport, is it an active transport or passive transport? See, it can be anything. See, vesicular transport is not dependent on the concentration gradient. Okay, it's independent. It's sometimes dependent on the concentration gradient, sometimes not dependent on the concentration gradient. Okay, so vesicular transport sometimes can be an active process, sometimes can be a passive process. Okay, now, what exactly is happening here? See, there is a transport of substances. Some substances are getting transported in the form of vesicles. Large particles are transported in the vesicles. Now, how many types of vesicular transport are there? There are two types of vesicular transport. One is called as exocytosis. Exocytosis means what? Means particles, large particles are getting out of the cell in the form of vesicle. By forming a vesicle, we are moving the substances out of the cell. That's called as exocytosis. And then what exactly is endocytosis? Endocytosis means by forming a vesicle, we are moving the substances into the cells. Inward movement is called as endocytosis. Outward movement is called as exocytosis. Okay. Now, let's see exocytosis. See, in order to perform exocytosis, let's say here is this cell. Now, this cell, okay, want to move some particles out of the cell. By forming a vesicle, now what it is doing, it is sending the substances out of the cell. By forming a vesicle, it is throwing the substances out. This is called as exocytosis. See, for this process called as exocytosis, you need to have certain proteins and some ions. So, the proteins which are involved in the exocytosis, they are called as the snare proteins, which was recently asked in the exam and repeatedly asked. So, snare proteins helps in which process? Exocytosis. And which ions are required for exocytosis? Calcium is required for the exocytosis of the proteins or the neurotransmitters or any substance for that matter. Exocytosis requires calcium as well as snare proteins. Now, you will start to have it out, sir. What exactly are these snare proteins? Now, let me tell you the examples of the snare proteins. The examples of the snare proteins are synaptobrevin, syntaxin, and SNAP25. Synaptobrevin, syntaxin, and SNAP25. These are snare proteins. Now, how exactly these snare proteins are helping in the process of exocytosis? How calcium is helping in the process of exocytosis? Let me tell you. I will give you an example, clinical example. Try to understand. Let me show you a neuron. This is the neuronal ending. Here, let's say this is the neuron. Which neuron, sir? Is it a sensory neuron, motor neuron? Let's say it's the alpha motor neuron. You know, alpha motor neurons are lower motor neurons which are coming out of the spinal cord, which helps in the muscle contraction. This is the motor neuron. Now, here, this area which I am highlighting in red color, let's say. What is this area? This is the muscle. Okay, muscle cell membrane. This is the muscle cell membrane, sarcolemma. So, can I call it as the neuromuscular junction, this area? So, this area which I am highlighting, this area is called as a neuromuscular junction. Neuromuscular junction. Now, see, here within the neurons, already there are vesicles. Okay, there are vesicles which contains the neurotransmitter. Which neurotransmitter? What are these dots? These dots are the neurotransmitter. Which neurotransmitter? This is the acetylcholine. The neurotransmitter, the neuromuscular junction is the acetylcholine. So, these dots which I am highlighting here, these are the acetylcholine molecules. Now, understand. See, this vesicle, a vesicle is having a protein. There is a protein attached here. What is the name of this protein? The name of this protein is called as a synaptobrevin. Synaptobrevin. Now, the synaptobrevin has to come and fuse with a syntaxin, let's say. Here, this molecule which I am showing you here, this is called as a syntaxin. Okay, synaptobrevin, syntaxin. See, this synaptobrevin and syntaxin, they should actually fuse so that these contents will be undergoing exocytosis. So, in order to release this neurotransmitter, let me put it this way. This synaptobrevin, it should actually come and it should fuse like this. Okay. So, what I am trying to say, how the neurotransmitters are undergoing exocytosis? Let me tell you. See, whenever the action potential, okay, whenever this action potential is running along the nerve membrane, whenever this electricity or you can say depolarization or the action potential, it is flowing along the neuron. This depolarization, once it's reaching the nerve terminal, once it is reaching the last part, that's the nerve terminal, this depolarization is going to open. The depolarization opens the voltage gated, means voltage sensitive, voltage gated calcium channels. So, now the voltage gated calcium channel will open because of this voltage. This is the new voltage, now depolarization. Because of this depolarization, the voltage gated calcium channels will open. So now tell me, what will happen to the calcium? The calcium ions will start to come into the neuron. The calcium is coming into the neuron. Now, what this calcium will do? The calcium will go and bind with the calcium is coming and binding with the vesicle. So now, because of this binding of the calcium, now this vesicle, it will move so that it will come and fuse with the synaptobrevin. It will come and fuse with the syntaxin. So synaptobrevin will come and fuse with what? Syntaxin. So who will be released? So there is exocytosis of the neurotransmitter. The exocytosis of the neurotransmitter acetylcholine is going to be released to the neuromuscular junction. Now what this acetylcholine will do? Now this acetylcholine, it will go and bind with its receptor. Here is the receptor of acetylcholine, which is a ion channel actually. This is the ion channel. Now what this acetylcholine is doing? Now this acetylcholine, it will come and bind with the receptor. The name of this acetylcholine receptor is called as a NM receptor. Nicotinic receptor on the muscle. NM stands for nicotinic receptors. Nicotinic receptor on the neuromuscular junction. NM receptor. So whenever this acetylcholine is available, acetylcholine is going to open this ion channel so that the sodium which is there outside, the sodium ions which are there outside the cell, sodium will start to come into the cell. The sodium is a positive charge which will lead to the depolarization of the muscle. It will cause a depolarization of the muscle. So you tell me, when a muscle is undergoing depolarization, the muscle will contract. The muscle is undergoing contraction, sir. So this is how muscle contraction occurs. This is how the muscle contraction is going to occur. So now tell me, what exactly is happening? Sir, whenever a neuron is getting depolarized, when the depolarization reaches the nerve terminal, depolarization opens which channels? Calcium channels. Calcium, have I taught you, it helps in exocytosis. See, calcium will go and bind with the vesicle. So the vesicle now it will move and bind with the vesicle. Will go and bind with the synaptobrevin, will go and bind with the syntaxin. So this fusion of synaptobrevin and syntaxin actually helps in exocytosis. Finally, what I am trying to say, these snare proteins called as a synaptobrevin and syntaxin helps in exocytosis of the neurotransmitters. Okay, for exocytosis, what is required? Calcium, synaptobrevin, syntaxins or snare proteins are required. Now you might be thinking, why this area is very, very important? Why I have told you this area is very, very important? Because let's say there is a child, okay, or a newborn, let's say. Now this newborn, okay, in certain parts of the world, there is this habit of giving honey to the child, believing that the child will speak early. So now this child is consuming honey, raw honey, or sporulated honey. Sporulated honey or raw honey. See, sporulated honey means what? It's a contaminated honey. Definitely there are some spores of the bacteria. Which bacteria? So this raw honey or the sporulated honey, it contains a bacteria called as Clostridium botulinum. See, this Clostridium botulinum is not only present in the raw honey or sporulated honey, it is also present in canned foods. Canned foods means the foods which are preserved in cans, like, okay, canned beans, canned peas, kind of canned olives. Okay, so whenever, even not only child, even you take canned foods, or a child who is taking a raw honey or sporulated honey, or contaminated meat also contains which bacteria? Clostridium botulinum. So today you are consuming this bacteria, you are having this bacteria into your body. Now this Clostridium botulinum, it will produce a toxin called as a botulinum toxin. Okay, so what it is producing? It is producing botulin toxin. Now this botulin toxin, do you know where it will go? The botulin toxin, it will enter into your neurons. And this botulin toxin, it will cause damage to, it will destroy synaptobrevin and syntaxin. Synaptobrevin and syntaxin are getting affected. Okay, synaptobrevin and syntaxin, they are getting affected. Now you tell me, the botulin toxin, what is the mechanism of action of botulin toxin? The mechanism of action of the botulin toxin is to destroy the snare proteins like synaptobrevin, syntaxins. So without this synaptobrevin, syntaxin, do you ever think this exocytosis will occur? The vesicle will release the neurotransmitters? No. Now in this baby, acetylcholine is not available. Acetylcholine is not getting released. Now without acetylcholine, do you think this sodium will come into the cell? Do you think the muscles will undergo contraction? No, the muscles are not going to contract. So the baby is going to have flaccid paralysis. Means muscles are not contracting, muscles are totally there, lose their tone. So there is total flaccidity. Okay, this is flaccid paralysis. This is also called as floppy baby syndrome. Okay, floppy baby syndrome. So now tell me, What is the mechanism of action of botulinum toxin? Botulinum toxin inhibits the release of presynaptic neurotransmitters. So what this botulinum toxin is doing? Botulinum toxin is inhibiting the release of what? It is inhibiting the release of neurotransmitter from which membrane? From the presynaptic membrane. So no acetylcholine, without acetylcholine muscles will not contract. Okay, the baby is going to end up in the flaccid paralysis. Especially how the paralysis is going to be descending flaccid paralysis. Descending means the paralysis will start from the upper parts of the body, slowly it will go down gradually. So descending flaccid paralysis. Okay, descending flaccid paralysis. Now, apart from this, what else you should know for your exams? Guys, listen. There is an autoimmune condition. Okay, there is an autoimmune condition where antibodies are produced against what? Voltage gated calcium channels. Same story. Whenever the calcium is not available, see, these channels are getting affected. So no calcium, without calcium the neurotransmitter release will be compromised. So same again, muscle contraction will be affected. So there is weakness in the muscles. So now what is that condition where antibodies are produced against the voltage gated calcium channels, right? Antibodies against voltage gated calcium channels. The condition is called as Lambert-Eaton syndrome. Okay, Lambert-Eaton syndrome. So Lambert-Eaton syndrome is a masthenic syndrome where there is weakness of the muscles because the neurotransmitter is not properly getting released because the calcium channels are getting affected. Next, in the same way, there is one more autoimmune condition, same autoimmune condition where antibodies are produced against the nicotinic receptors. See, now the antibodies are produced against this NM receptors. So, do you think acetylcholine can come and stimulate the muscles? No, not possible. So, what is this condition called as? Whenever the antibodies are produced against this receptor, that is NM receptor, that condition is called as, right, antibodies against NM receptor or acetylcholine receptor, you can say. That condition is called as very, very important, myasthenia gravis. Myasthenia gravis. Myasthenia means, myo means muscle, asthenia means weakness. It's a grave weakness, severe weakness of the muscles, myasthenia gravis. So here, three things I have explained you. Three things. One is what is the mechanism of action of the botulinum toxin, and what is the pathophysiology lies in the Lambert-Eaton syndrome. The Lambert-Eaton syndrome is a problem with the calcium channels, and myasthenia gravis is a problem with the nicotinic receptors. See, myasthenia gravis is very, very important for your exams. How the question will come? They will say there is progressive weakness in a day. In the morning muscles are okay. Okay, everything is okay. But with the time, so the more the muscles you use, more weakness will come. There is a progressive weakness. Progressive weakness of the muscles. Okay, progressive weakness of the muscles. But what about the Lambert-Eaton syndrome? See, in Lambert-Eaton syndrome, initially there is muscle weakness. The more you use, the better they becomes. Okay, progressive betterment. Progressive betterment means in the morning there is weakness, and with the time the muscles are getting better and better. Okay, the myasthenia decreases. So this is the one important way to differentiate between whether it's a myasthenia gravis or Lambert-Eaton syndrome. Okay, so with this, what we have completed? We have completed the exocytosis. So finally tell me guys, exocytosis, tell you what is the required? Exocytosis requires snare proteins as well as calcium. Snare proteins as well as calcium. How to remember? See, snare proteins, how it is sounding like? See, snare always sounds to me like a snare, snake. Who is a snake in your life? Your ex is a snake in your life. Just remember like that. Okay. So snare proteins helps in exocytosis. Exocytosis. Okay. Now, then what about the endocytosis? Just like for exocytosis, you require some proteins, right? Like snare proteins. In the same way, for endocytosis, you require certain proteins like clathrins and caveolins. This was also recently tested. Clathrins and caveolins, they will help in the formation of a vesicle in order to move the vesicle into the cell. Okay, clathrins, caveolins. And how many types of endocytosis are there? There are different types of endocytosis like phagocytosis, pinocytosis, receptor-mediated endocytosis. These are the different types of endocytosis. For example, what is the example for the phagocytosis? Let's say there is a macrophage or a neutrophil eating the bacteria. Okay, macrophage, what it is doing? See, there is a bacteria outside the macrophage. It is extending the cell membrane and forming a vesicle around the bacteria, eating the bacteria. So this is an example of phagocytosis. Phagocytosis means macrophage eating bacteria, or a neutrophil eating the bacteria. That's an example of phagocytosis, means cell eating, cell eating. So cells not only eat, they also drink, sir. Okay, they also drink. So whenever the cell is getting dehydrated or in any other time, if a cell is taking the extracellular fluid, see outside the cells there is extracellular fluid. If the cell takes the extracellular fluid in, that is called as a cell drinking. Cell drinking. Okay, also called as a pinocytosis. Pinocytosis. Phagocytosis means cell eating. After eating, it will drink. Just like you, after eating, you will drink, right? In the same way, cell first it will eat, later it will drink. Like phagocytosis and pinocytosis. Both are examples of endocytosis. Then what is receptor-mediated endocytosis? See, receptor-mediated endocytosis, example, let's say, here there is a myocardium. Okay, here there is a myocardium. Now, whenever there is too much sympathetic activity, or whenever there is too much epinephrine or norepinephrine, maybe conditions like pheochromocytoma, there is excessive amount of epinephrine is available. Now, whenever there is excessive sympathetic activity or excessive epinephrine is available, now what this epinephrine will do? The epinephrine, it will come and unnecessarily, this unnecessary epinephrine, okay, what it will try to do? It will try to stimulate the receptors unnecessarily. So what will happen to the heart rate? There is increase in the heart rate. So tell me, is it good or bad? Bad, no. See, this high levels of epinephrine is because of some tumor called as a pheochromocytoma. Because of this excessive amount of epinephrine, there is excessive stimulation happening. Because of that excessive stimulation, there is more heart rate, which is not good, definitely bad. So now in these conditions, do you know what the cell will do? Cells, now this part of the membrane, from here to here, this part of the membrane, do you know what happens? Now this part of the membrane will start to invaginate. It will start to invaginate. Let me show you that. That part of the membrane got invaginated, like this, invaginated. And what happened to the receptors? Now the receptors are now taking into the cells. Now they are moving into the cells. So finally what will happen? Again the membrane will form. Here the vesicle was taken in. So again endocytosis. Means what we are trying to do? We are trying to remove the receptors from the surface of the cell. Okay. Now these receptors were taken into the cell. So what happens to the less number of receptors? Less number of stimulation. Less amount of stimulation. More amount of receptors, more the sensitivity. Less amount of receptors, less the sensitivity for the cell. So this is called as a receptor-mediated endocytosis. Okay. So with this, successfully what we have completed the topic of vesicular transport, which includes the endocytosis, exocytosis. Exocytosis requires snare proteins. Endocytosis requires caveolins and catherines. Okay. Hope the video is helpful. See you in the next video. Thank you.